In this video, we will be looking at patterns of epistasis, specifically single recessive epistasis in the color of roses. Before we begin talking about epistasis in our pathway, I want to discuss some terms that you should know. A gene is a unit of heredity that is passed down from a parent to offspring. An allele is a variation of a gene found on the same place on a chromosome. A locus or loci is a point on the chromosome that codes for a specific trait. A chromosome is a molecule that holds the genetic material of an organism. A genotype is an organism's collection of genes, which are shown in their lettering. With homozygous genotypes, it is two of the same allele. With heterozygous genotypes, there are two different alleles being expressed. A phenotype is an organism's expression of their genotype, which in our case will be white, pink, and red roses. Some other terms that you should know are a Punnett square, which is a representation of possible genotypes found when crossing two parents with specific genotypes, which you can see below in the cross between a big A little a parent with another big A little a parent. In a dihybrid cross, you are looking at the mating between two identical hybrid individuals for two traits, being a cross between two big A, big B, little b parents. This cross results in a phenotypic ratio of 9:3 to 3:1, and this will be important to remember throughout the rest of the video when we begin talking about single recessive epistasis. Now I want to give you a brief description of epistasis, and more specifically single recessive epistasis. Epistasis is when the expression of alleles at one point on the chromosome can mask or change another allele at another locus. This is an interaction between genes, not alleles, and it is important to note that multiple genotypes can lead to the same phenotype or phenotypic expression. Another definition that is important is epistatic, which is a gene that masks the action of another gene. In single recessive epistasis, a homozygous recessive trait at one locus masks the expression of a second locus on the chromosome, meaning that little a is epistatic to big B and little b. This pattern of epistasis results in a phenotypic ratio of 9:3:4 due to the grouping of two phenotypes. In our pathway of single recessive epistasis, we have a colorless precursor, our pink intermediate, and our red end product. At enzyme A, you can see that if there is a homozygous recessive, there will be no enzyme A and the roses will remain white. But if there is a dominant allele, A, then the rose will become pink intermediate. Similar to enzyme A, enzyme B will have a rose remain as a pink intermediate if it is homozygous recessive little b, but if it has a dominant B allele, then we will reach the red end product. In our roses, you can see that the little a locus masks the big B and little b loci. To better understand this pattern of epistasis, we can perform a dihybrid cross. This 4x4 Punnett square shows how the possible genotypes for the offspring of two identical hybrid parents. As we are focusing on the single recessive epistasis pattern, meaning that we should observe a 9:3:4 phenotypic ratio in our roses. Looking at our pathway and the different genotypes in the Punnett square, we can see that there are 4 genotypes that result in our white or colors precursor, 3 that show our pink intermediate phenotype, and 9 that express our red end product. This creates our 9:3:4 phenotypic ratio. Alternatively, we can look at the cross with random genotypes as shown in the Punnett square. In this cross, there are 8 genotypes that result in white roses and 8 that will result in red roses, giving us a 1:1 phenotypic ratio in this alternative cross. And here are my references.